So in the video before we spoke about ADH and how it's released when one is dehydrated, or actually more correctly when one's blood concentration or blood ion concentration rises above the homeostatic norm. Now we're going to see what ADH actually does, and it shouldn't take too long, as a lot of this is actually sort of extension for things you will need to know in a different setting. So ADH's main action is on the collecting duct, and the collecting duct has this sort of anatomy, this structure where you have a vasa recta, which is one of these blood vessels. So that's our vasa recta running alongside it. And when we are dehydrated and when or our blood concentration level is high, we require more water reabsorption from our collecting duct. We've got some water reabsorption happening at the PCT and even some happening at the loop of Henle, and a little bit at the DCT, but the main bit where we alter how much water we're reabsorbing is here. So what happens? We've got ADH traveling through the blood, okay? It's been released into the blood by the pituitary. Some of it will come and it will bind to our ADH receptor. We bind to our ADH receptor, and that causes this G protein to swap its GDP for a GTP. So GTP now rather than GDP, and this is just like ATP versus ADP. It's just the exact same thing, but with a guanine rather than an adenosine. They exist. Okay, so this is something you haven't met before, but it's important. Once this GTP binds, this G protein detaches from the receptor. So this makes this a GPCR, a G protein coupled receptor. This G protein will then go and bind to adenylyl cyclase, which is an enzyme you may have met before. Adenylyl cyclase is then activated, and it turns our old friend ATP into this signaling molecule called CAMP, which is cyclic adenosine monophosphate. This CAMP will then sort of diffuse around the cell, and it will start binding. It will start binding to these little vesicles, which contain aquaporins in them. So they're sort of studded. And as they do, what they end up doing is basically exocytosis, and they will fuse with this membrane. This membrane is usually impermeable to water, as we see here. But now, what ends up happening is you get all these vesicles fusing, and you've now got all these water channels, these aquaporins, allowing water to go down its water potential gradient, because this area is saltier or got a higher concentration of sodium chloride, if we're being very scientific and clever, than this area. So this area is dilute in comparison. The big question is, how does this get salty? We haven't spoken about that, and that is the role of the loop of Henle.